Alzheimer's disease is becoming more common as our population ages. Before we begin to think specifically about Alzheimer's disease, let's think about neurodegenerative diseases in general. They can be classified according to the abnormal proteins that accumulate in their cells or the other specific pathological abnormalities. Thus, we can consider Alzheimer's and Pick's disease as tauopathies because they both involve the accumulation of the abnormal tau protein. Similarly, synucleinopathies, Lewy body disease, and Parkinson's disease involve accumulation of abnormal synuclein in the cells. The accumulation of these abnormal proteins kills the neurons. In Huntington's disease, we have another abnormal protein caused by a CAG repeat. Similarly, in CJD, the abnormal proteins involved in amyloid production accumulate in cells. Alzheimer's disease is the most common form of dementia, accounting for over half of all cases. The prevalence of Alzheimer's dementia is low in people less than 65 years of age, but there is a rapid increase such that in people aged 85 years or older, the prevalence is between a quarter and a third of all adults. Inherited Alzheimer's disease is very rare as an autosomal condition. The people who have the autosomal mutation in either the amyloid precursor protein or presenilins 1 and 2 usually have an early onset before the age of 65. Most mutations in the APP and presenilin genes increase the production of a small protein, which is the main component of the senile plaques. Sporadic Alzheimer's disease has also been shown to have a significant genetic background. A large population-based twin study showed that the extent of heritability for the sporadic disease is almost 80%. We know that the ApoE4 allele is important in Alzheimer's disease, as between 40 and 80% of people with Alzheimer's disease possess at least one of these alleles. One copy increases the risk of disease by 3 times, but if a person is a homozygote for the ApoE4 allele, then they have 15 times the normal population risk of getting Alzheimer's disease. Other risk factors in getting Alzheimer's disease include being Caucasian, women, and having the risk factors associated with vascular disease, such as hypercholesterolemia, hypertension, atherosclerosis, smoking, obesity, and diabetes. It is also increased in people with head injury. Perhaps the latter two decrease the brain's reserve, and so early Alzheimer's will give more severe symptoms. Alzheimer's disease is a protein misfolding disease caused by plaque-like accumulations of abnormally folded beta amyloid and tau amyloid proteins in the brain. Beta amyloid is a fragment from a larger protein, the amyloid precursor protein. The amyloid precursor protein is a transmembrane protein which is critical to neuron growth, survival, and post-injury repair. This series of illustrations shows how beta amyloid is generated from the APP module as enzymes cut it out of the disassembling protein. The abnormal beta amyloid clumps together and forms plaques. We can think of these plaques as inhibiting neurotransmission. The senile plaques are seen on H&E stain as diffuse areas with a dense core, as seen in this histology picture from a person with Alzheimer's disease. The cells surrounding the plaque are mostly astrocytes, with the red areas being capillaries. The plaques are much easier to see if a silver stain is used. Here we can see a central dense core in the plaque with surrounding abnormal silver staining proteins. Alzheimer's disease, while characterized by the accumulation of beta amyloid, is also characterized by the abnormal accumulation of tau protein. Every neuron has a cytoskeleton, an internal support structure partly made up of microtubules. These tubules act like tracks, guiding nutrients and molecules from the body of the cell to the ends of the axon and back. A protein called tau stabilizes the microtubules when it's phosphorylated. In Alzheimer's disease, tau becomes hyperphosphorylated. It then begins to pair with other threads, creating neurofibrillary tangles and disintegrating the neuron's transport system. The process is illustrated in this drawing, where when tau forms tangled clumps, the microtubule subunits start to fall apart. When we look down the microscope at the brain of someone with Alzheimer's disease, we can see the neuritic, also known as senile plaques, and neurofibrillary tangles. The neurofibrillary tangles are the tau protein tangles. There is always significant loss of neurons in Alzheimer's disease, in addition to shrinkage of large cortical neurons. Many investigators believe that loss of synapses in association with shrinkage of the dendritic arbor of large neurons is the critical pathological change. Here we have a microscope picture of someone with Alzheimer's disease. In this, the neurofibrillary tangle is illustrated. The neuron in the center of the picture is of a much darker hue than normal and is carrot-shaped. This is because it is filled with neurofibrillary tangles. Again, we can see the changes much better on silver stain. You can see the round senile plaque, and then to the left of this a carrot-shaped neuron which has got neurofibrillary tangles in it. Macroscopically, the brain in Alzheimer's disease also shows marked changes. On the left is a normal brain. On the right is an illustration of a brain with Alzheimer's disease. There is shrinkage of the gyri and expansion of the sulci, particularly in the forward part of the brain. In addition, there is extreme shrinkage of the hippocampus. The atrophic brain leads to enlarged ventricles. So, the characteristic macroscopic features of a brain with Alzheimer's disease are marked atrophy with widened sulci and shrinkage of the gyri. The occipital pole is relatively spared. The cortical ribbon may be thinned, and ventricular dilation is apparent, especially in the temporal horn due to atrophy of the amygdala and the hippocampus, which are some of the most severely affected parts of the brain.